Thanks everyone for joining today's meeting. So let's go and start it because we have 3:30. We have other people joining in. So just go quickly with the normal procedure of our meeting. So today our 7th meeting. So I will ask everyone to, can you see my screen? Okay. Okay. And before start, as usual, I would highly encourage you to be according to our ground rules. That means we support all our whole opinions and critics idea, not the people, and we want to hear everyone's voice. For this meeting, I created a Padlet discussion because I thought some people may not be able to join our meeting. So in case if you find you want to ask a question, you can post your question under each category of the tablet I created for the each section for the each discussion. In addition, when speakers are presenting their research, if you have a question, you can post it in there or wait until they finish, then you can ask your question. Okay, either way is fine, but I hope we will able to answer all questions. If not, please post your answer in the tablet. I think all our 3 speakers, Amanda, Gil, and David, can answer later on if they was not able to find the time to answer during this meeting. Okay, so I will go with the order that I noted in the agenda, and I will ask Amanda to go first. Are you ready, Amanda?
Yeah, I'll get my screen share set up here.
Okay, I will stop sharing.
I feel like this is never not awkward. No matter how long we do this. Okay, do you see my slide or my notes?
Slides.
Great. Okay, so howdy y'all. I think I met almost everybody at the boot camp, but to reintroduce myself again, my name is Amanda Stahlke. I'm a postdoc with the USDA Agricultural Research Service, a computational biologist postdoc, and I work remotely in Western Colorado, but Anna Childers is my supervisor, and I am technically based out of Beltsville, Maryland. My role in the BPRI is on the assembly team, and I think I'll interact with the transcriptomics team a bit, but I was brought on as part of the Ag 100 Pest Initiative, which is the initiative that Dr. Childers heads to provide high quality genomes for agricultural pests, including locusts. So for my 3 to 5 minutes here, I wanted to provide some opportunity to ask for you all to ask questions about the genome assembly process and provide some training on how that process works and talk about where we're headed with all of it. So genome assembly in a nutshell. This is what we did to get these beautiful genomes. We squished up Schistocerca and put it into a PacBio SQL and generated long reads shown by the gray bars here to assemble contigs. So when you hear contigs, that is just from the long reads, and they have no gaps between them. We generated a linearized consensus primary assembly. And if there are many other ways to do this, so if you have questions about like, well, why didn't we generate completely haplotype phased assemblies, that's a totally valid question. We can go down that path, but we generated a linearized consensus primary contig assembly from the PacBio reads and alternate haplotypes to complement for each species. Then with another locus, this is important, so 2 separate samples for all of these genome assemblies, we generated Hi-C data, which capitalizes on the chromatin capture of the chromosomes themselves to scaffold those contigs into what's called scaffolds, but in this case, they are chromosome length. And so the way this algorithm works is that you use the density of the Illumina reads shown in red that correspond to where the chromatin capture occurred to identify where the contigs lie in proximity and orient them correctly. And so that's genome assembly in a nutshell. And that takes like 15 to 20 people for each of these species, lots of trial and error to get the highest quality extractions, and then a lot of time to make the algorithm work as well as it can, and then do the manual curation. But with all that work, we now have these unprecedented genomes compared to all other insects in NCBI. So each of these points here, I downloaded the metadata from NCBI, and the black are all other insect genomes. Then we have the previous locust assemblies on the bottom right with these huge genomes, and then we have our BPRI locus up here. And I would say we should be really, really proud of this, but this would not be possible without the current sequencing technology. Like, that's really what's doing the heavy lifting here. And I would say like that is what made it possible more than ingenuity, but we should be proud of it. But genomes don't provide insight alone. I know we've been very proud of our genomes, but I think it's important for me to acknowledge that they are just foundational components to support the rest of the BPRI. That being said, they can generate hypotheses and test some hypotheses, especially in coordination with all of these other components that the rest of the BPRI is providing. As a starting point, my role for this project is to look at the repetitive content or transposable elements in these genomes. And since 70% of these 9 gig genomes is repetitive content, there's a lot of work to do. And I just want to remind folks that TEs are these really bizarre kind of parasitic elements of the genome. This infographic here is displaying 10 things you should know about transposable elements, and I just want to point out a couple of them. So they take different forms and shapes. They're not randomly distributed throughout the genome. And then they have quite a few different kinds of implications for phenotypes that we might be interested, including modifying regulatory networks and altering expression and repression. Oh, and they can rearrange the genome. So with all those things in mind, I'll show you what the repeat content looks like for genomes that I have visualized so far, including these 4 species: Americana gregaria, Piscifrons, and Cancellata. On the right side, these are different categories of transposable elements, and there are a lot of them. We can talk about that if you would like, but the pie charts show the repeat content respective to each of these species. And what you should see right away is that this TC Mariner is, represents the plurality of classified repeat in all 4 Schistosarcus species represented here, as well as the other 2 that I'm not showing. What is most interesting to me is that if you look at the age of the repeats measured by this Chimera 2-parameter divergence, it's just a nucleotide sequence similarity measure, they all have different shapes. So that means that all of the repeats have been moving differently in these genomes, which may contribute to differences in their phenotypes. So moving forward, I'm trying to help contribute to the BPRI overall. Some tests to see how TEs contribute to plasticity of phase polyphenism. And I have a lot of work to do still, including additional curation or classification of these repeats. I have estimated divergence for all species now, but not visualized all of them. And right now, I'm really spending most of my effort trying to associate the physical distribution relative to the genic and chromosomal regions. We already know that TE activity can influence gene family evolution. So I think it could be an important player in phase polyphenism, but we'll have to bring in other data types like the gene expression and attack-seq, et cetera. So I'm excited to work with all of you to see how the genomes themselves at a base level are contributing to the phenotypes that we're interested in. And I look forward to all of that. That's all I have.
Thank you. So anyone have questions for the Amanda or clarification, or if you want to get more information about her research? Okay, David, go ahead.
Yeah, I have a question. Could you explain how that age metric for those transposons worked again? You're muted, by the way.
How did I do that? Okay. So the Chimera two-parameter divergence, it's a classic evolutionary distance measure. You'll see it in like the classic PopGen text. But the idea is basically like we expect some transitions to occur at a different rate than others, like A's to T's. And so it's using a model-based estimation of sequence divergence between the repeat classes. Does that make sense?
Yeah. Shouldn't the Gregaria one be like the one with like the greatest age length then? It didn't like really seem like it from that graph, at least. You're muted again.
I don't know if I'm clicking that. Okay, so tell me why you think that, because.
Yeah, so from my understanding, Gregaria is like the most basal species that we're sequencing, right? So it theoretically should be closest to the greatest common ancestor of all those Schistosarcus species. And so the transposons will have moved around the most or something, I don't know.
But that is an excellent neutral expectation if there is no selective pressure or other processes at play. And so like that is exactly the kind of thinking we are bringing into testing these kinds of hypotheses. Like, okay, here's a neutral expectation, here's how many repeats we expect and how old they should be. And now, you know, what lies outside of that model-based estimate? So that's a great question.
Thank you. I'm sorry we cannot get more questions. So please feel free to add your question to Padlet. Amanda, in meantime, you can check and answer. Okay.
The more Amanda talks, the less I have to. So that's why I'm asking those questions.
Okay. So Gil, can I move forward to Gil's presentation?
Yep. Let me start screen share. Sorry, where's the screen share button again?
In the bottom. You can find
there it is, the one
with
highlighted
the
one.
arrow to up, kind of.
Yep, I see it. All right. Okay, can you guys all see?
Yes.
Okay, so hi, I'm Gil, also at Baylor College of Medicine in the Gabbiani lab. I do electrophysiology, and I've been testing the effect of a drug against deactivating potassium currents on our kind of PET neuron. So this is the slide that I presented at the BPRI training, but I'll just go over it one more time. So we take a locust and we cut it up so that the optic lobe is exposed. and the eye is facing a TV screen, then I get a stain of the LGMD with an intracellular recording. So this is generally the shape of the LGMD. It's got this long neurite that goes into the brain, and coming off of it are multiple dendritic fields. So this main big here is called field A, and it's as the eye. And then here you can see two more little neurites going off. This one is going to field B, which is directly underneath field A. And this one here is going to field C, which is kind of conical shaped and goes straight down into the optic lobe. Here on the right, I have a video of the stimulus we present. It's just a black square that appears as an object moving at a constant velocity would. So it appears to get bigger as it comes towards the screen. On the top trace is an intracellular trace, and the bottom is the extracellular DCMD trace. The DCMD is a neuron in the nerve cord that has a one-to-one spiking parity with the LGMD. So just by having that extracellular recording without having to hold an intracellular recording while I do the whole experiment, which is very difficult. So these are summaries of the data to various stimuli. So this is the stimulus that I just showed. It's a black square that just on a white background that looms expanding outward in all directions. Up at the top, I have a mean instantaneous firing rate. So looking at for each animal, what was the frequency of firing in a small moving window across the time relative to collision. So 0 is when an object moving like that would theoretically intersect with an animal. And by calculating this on average across multiple trials on multiple animals, I get this trace that shows spiking ramps up kind of steadily to a peak that happens right before collision. Blue here is the control experiment, and the red is after I apply 4AP. You can see there is a pretty clear increase, mostly right at the end, like right in that last couple milliseconds before collision is where most of the increase is. Down on the bottom here, I have graphs. Each circle or each line is one animal. And this is kind of just showing some of the variability in the data I've got. So the on the left is the maximum IFR. So whichever point for each animal had, the highest peak, and showing the difference between control and 4AP for that, as well as the total number of spikes that happened during this relevant time frame. So as you can see from this is kind of, so first I want to say that the black coherent loom, as well as the black scrambled loom I'm going to show later, Richard Duell, a postdoc in the Gabbiani lab, has already showed the effect of 4AP on this. And so this is a nice kind of confirmatory that the amount of increase in firing rate from this drug is in line with what we've seen before. However, as you can see, there's kind of a lot of variability among these animals, and I'll talk about that later. But yeah, this variability in the firing rate is a little unusual, as well as having some animals that actually decreased firing rate in response to the drug. So now I'm moving on to some new data. This is the exact same stimulus I showed y'all, but it's color swapped. So it's a white square looming on a black background. And it also looks like there was a significant increase in firing in this IFR mean for the white, also pretty variable. Generally fewer spikes for a white loom than a black loom. And we see that there is, in fact, a 4AP increase. Now, this is a black scrambled loom. So this is a stimulus that has the same overall luminance change as that black loom that you saw in the video, except the spatial component is randomized. So instead of having it start at the center being black and kind of expanding around, you have kind of what looks like TV static, except it starts all white and finishes all black. And so this has generally lower spiking than a coherent loom, but there's a greater effect of 4AP on the increase of spiking. So you can see that that's also in line with previous data. There's a much bigger jump because of 4AP for the black scrambled loom. Then doing the same thing for the white loom, this was a little less clear. The stimuli just tended to have pretty varied responses, more so than normal. So this one is more questionable whether 4AP had an effect. So this is just the summary statistics for each of the individual stimuli on their own, whether the rate, so in columns 1 and 3 are the ratio, so drug value divided by control value. And you can see that there's basically a significant increase for all of the stimuli. The white scrambled loom, the IFR max wasn't significant, but the number of spikes was significant. I don't know, but that's kind of in line with what it visually looks like. But comparing the difference in effect across the two, so this one here comparing the black solid loom with a black scrambled loom, this is already published or already known, already published by Richard and the Gabbiani lab, that there is a significant difference, that the black scrambled stimulus has a much greater effect from 4AP. But we didn't see a consistent difference in effects in basically any of the other stimuli. The black scrambled and white scrambled had significance for mean spikes, but not for the IFR max. And then just going back to look at what the values were. So yeah, you can see going from black to black scrambled, there's kind of a big jump. The white stimulus had similar statistics to the black scrambled, and the white scrambled had similar to the black solid. So what I think is that there were a couple of animals in there that were habituating more during the experiment, or maybe weren't that healthy to start with. So what I'm planning to do is go back through those data and kind of see if there's a good reason why any of them should be excluded, although I don't really like doing that after the fact. So mostly what I'm doing now, and this is kind of getting into the future stuff, is I'm just going and adding more replicates to this. Oh, sorry, and I should have mentioned, all of these data are for puffing 4AP to field A. So that big parasol that was there in the beginning. Yeah, so I think that the reason why it's kind of hard to interpret like a non-significant result, but I think that a lot of this is just pretty wide variability in kind of the response to all of these stimuli. So I'm just going back and adding replicates to field A. I've also done a couple of experiments puffing to field C, but I didn't feel comfortable doing statistics on it yet. I only have 4 animals for that. And we're also going to try some iontophoresis, where you use current, like you would be injecting current into a cell, but instead have that move the charged drug. And we're also going to do some targeted intracellular recording. So like I said, I like to use the extracellular recording because it's easier to do, especially when you're doing something like mechanical, like puffing drug, but eventually I'm going to have to do some intracellular recordings. while I populated the drug as well.
Gil, I'm sorry I
And
had to interrupt.
yes.
Time is up. So do you have any more slides?
Nope, that was it. That's it. Targeted intracellular recordings and some other drugs for other channels later.
Many thanks, Gil. Thank you very much. So any question for Gil? Seems like it is silence. If you have a question or if you need any answer, please add to the Padlet, then Gil can answer. Now we are going to David. Can you? I hope you can share the screen. Oh. Yes, we can see. Thank you. We can't hear you.
All right. Can you hear me now? Hello? They can hear you?
Yes.
Okay. Okay. All right. So real quick, because I don't have much time. Right now, I'm kind of focusing on doing transcriptomics on the optic lobe. So the first thing that I did, and you guys probably saw this already in the presentation during the big BPRI meeting, was that I just did some differential gene expression analysis on the LGMD neuron, which is that big main neuron that Gil does experiments on from his previous presentation. Right now, the analysis is kind of a little bit of a rough stage because we don't really have like a super good control to measure the single cell LGMD against. So we don't have like as many significant genes as we would like to see pretty much. And whoa, okay. So basically, I've been learning how to do all the steps necessary in order to create a cell atlas of the optic lobes and maybe even eventually the whole brain. We'll kind of see how that goes. So, you know, here's me mutilating one of those poor locusts, but I've actually been able to pick it up pretty well. And so I've been able to extract some optic lobes. And so I've kind of moved on to the next part, which is called MAT-QSeq. And MAT-QSeq is a single cell sequencing protocol that was developed by Chuck Zong's lab, just here at Baylor College of Medicine. So I've been going up to his lab to kind of learn how to do that protocol. And what you're seeing in the picture here is what it actually looks like under a microscope. So you have, you know, various tubes that are putting in different fluids needed for the reaction. So if you guys can see my mouse over here, the top left is where the beads are that contain the DNA barcodes that will actually label each single cell. The cells, the nuclei come in through this tube over here, and then over here we have the reaction solution. And then finally the bottom tube over here is where the oil comes in that will actually separate out these droplets from each other. So yeah, there we go. Any questions?
Yeah. How similar is the droplet technology to like a 10x is what I'm most familiar with? Is it kind of the same technology?
Yeah, so I'm actually not familiar with the 10x genomics protocol at all. And I've only been learning the MAT-QSeq like as of like last week. But the reason that we're using this one instead is A, it's in-house, so the costs are smaller, and B, when they published the paper, they actually demonstrated that they had greater sensitivity and less noise, less technical noise than 10x Genomics and SmartSeq and some of those other mainstream single cell technologies out there. So
I would also like to add that this one allows for like sequencing of fixed sample. So pharma has fixed sample, so that's also another advantage.
Yep. Thank you, GI. GI is a lot more familiar with this stuff than I am because she's been in the lab for a lot longer than I have. So
Thank you very much. Thank you very much for all of you for presenting your research, and it was very insightful. But honestly, I'm very kind of like a nursery student for this, your whole area. I cannot kind of grab all the sense, but I will try my best to understand what you guys doing. So thank you very much for your effort for trying to put together research presentation and doing that. So for the next part, I noted that because we are now moving forward after all our trainees present our research, we are going to forward with the next activities or how we're going to move forward with this monthly meeting. So today we have 3 guests for our meeting, Fabioso, Hojun, and Spence, on behalf of our BPRI leadership, joining our discussion from now onwards about bootcamp and how we want to move forward. So please allow me to share my screen and start the discussion. So the first one is about our bootcamp. So we had that meeting in May, but we need to review back for how we did well, especially how trainee feel about this meeting, and get your feedback about what went well and what are the opportunities that we can improve. Why we are doing that is we are going to have our 3rd year symposium next year in Baylor College, or Arizona State University. I don't know exactly where it is. I forget exactly where it should be holding. But if you have any opinion or any suggestion that we can improve our bootcamp, please feel free to share. And I will open the discussion for everybody to share your opinion. So please go ahead. Thank you. Anything at least went well?
I thought I really appreciated the boot camp. Coming from the outside, like I didn't know anyone at all, and I got to interact individually with almost every single person I think that attended the boot camp. So I thought like the opportunities for genuine interaction were really super, really good, like stimulating conversation in the like breakout sessions. And it was obvious that the entire group was interested in each other's perspectives. I think the whole thing went really well. It's hard to pick a single component.
Thank you, Amanda. I noted in the, like, just soon after the survey that I've given to you, some people say that the final discussion is kind of a little bit unfamiliar for the trainee side, because most of the PI was talking about how the way forward direction about selecting the project. So what your guys' suggestion on how we can improve such kind of way forward discussion and how you guys can contribute? Or in other words, how we can do it differently, in your opinion, if you have any idea? Because that was our first time, actually, Hojun and I, an executive committee meeting. We were struggling to how to do it a better way. So we also learning.
One possibility that I'm thinking was maybe to do kind of parallel sessions and then come together, so that I mean, obviously when everybody's together, it's kind of more difficult for everyone to kind of talk or share their ideas. So that's one thing that comes to my mind. But...
Thanks.
Yeah, I think as like a trainee, possibly some of the way forward stuff is a little bit big for what we're thinking about, I guess, because we have like smaller projects or smaller things in mind. So sometimes when we start talking about like really big ideas, it feels hard to like see how each of us fits into that, maybe. I don't know.
Yeah, that kind of makes sense. But that's like actually good to see also. It's a good exercise to kind of try to work out the big picture. But yeah, so clearly it requires, and we that's also part of our job to think about that. So
Yeah.
Obviously we're kind of more practiced in that.
Yeah.
But it certainly is good also for trainees to kind of learn that kind of stuff.
Mhm.
Yeah, I agree. I think like I was thinking that for that kind of like discussion, it was kind of beyond my understanding and ability to like contribute to it, but like this was the first time I was like seeing that discussion happen, what like the future of a collaborative effort, like the way to have that discussion. I was like, oh, so this is the way that like people plan and talk about this. So like I wasn't an active participant in it, but just, you know, like learning how that discussion can happen, you know, between PIs was, I don't know, I thought that I got what I needed to out of it.
I feel like the way forward discussion, like there was a lot of pressure to like make a decision and decide on a question, and it felt a little artificial to me. Like that's not really what happens. Like you stew on things, you have that big discussion, and then you go home and write emails and draft documents. And so like it felt like a lot to process and try to like commit to something when like that's not really realistic. Like that's not what's going to be the way forward necessarily. So I would suggest like one thing we could try for next time is to present some ideas, but then go home with them and come back again after a few days and after you've all had a chance to like do a little bit of research and maybe write some text. That's partially just how I process things, but it seems crazy to try to like identify the way forward in such an overstimulating weekend.
Yeah, I agree. I was, you know, a bit naive in thinking that we could actually, you know, make a decision. And what I kind of got out of it was there are many ideas, and everybody's understanding of, you know, the project, or even, you know, what locusts are and the biology of it. The level of understanding varied quite a bit. We can only have stimulating, you know, meaningful, integrative, you know, direction settled when everybody is sort of at the similar level, thinking about all those things a lot. And for a lot of students, it was the first time hearing from other labs. And then, you know, not only that, not just other labs, but a very different discipline, right? Like, I don't know what neurobiology is about, but, you know, now we have to connect. And those are all difficult, but at the same time, I think it was a kind of really good opportunity to think outside of your box, right? You're kind of forced to talk to somebody other than, you know, somebody who does what you do. So in that sense, I think it was productive, and you know, I think that the way forward is really, you know, sort of continuous process, right? But at some point, we do need to make a decision. Just that it couldn't be that moment, and so, you know, I get that you had a like sense that it was a bit artificial, and it was, right? and there's no denying that. So, but you know, I appreciate the feedback.
I really like the idea. I'm sorry.
David?
Yeah, go
I'll
ahead.
go
Amanda, go
after
ahead.
you, Amanda.
Amanda, you can go ahead.
Okay, I really like the idea of getting together a review article. I thought that was really could be a very productive way to bring all of the pieces together and like develop that common language. And I think just that exercise could help us move forward together.
Yeah, it will happen. It's either me or Ariane who need to pull a trigger. So it will happen.
One suggestion that I think I already gave to some of you guys after the meeting is before we went to the BPRI training bootcamp, they had us fill out this survey that one of the questions was like, what are you like most interested in learning more about? And then there was like the different aims from the proposal. And I thought that, you know, what was going to happen is we were going to have kind of like workshop style like stations, and then you can go like choose some of the things that you want to learn more about and go and attend like a little workshop where, you know, whoever is kind of in charge of that aim will teach you a little bit more about what's going on. You know, because I think I selected like I was interested in learning more about like the genome assembly and like the transcriptomics and some of the other things, right? So I think that would be a good way for everyone to you know bring themselves to the same page, you know, because I might already know a lot about this topic, but I want to learn more about this other topic that is part of the BPRI. So that's just a thought.
I think something like this could be organized. I mean, and especially if we are having quite a lot of faculty there, we could have, you know, a few faculty together and answering questions, kind of grouped by kind of expertise or so. Yeah, I think that would be good.
I think this goes along with the review paper, but it would also be like nice to provide like some foundational papers before we all get together.
Yeah, so at one point I was thinking that, but I don't know if that's a good idea or not, is maybe we could organize a journal club, ask all the PIs, you know, what is the kind of the important paper that they think everybody should have read. Because some of the papers that were mentioned, I think I haven't read either. And maybe have people present them and maybe have the PI that proposed the paper to be present so that he or she can answer questions. As a way of kind of introducing everyone to all those topics. But of course, it kind of adds more work. And so, you know, I was not sure if the trainees would be interested. I think it would make more sense if a trainee presents one of those papers. And some faculty that know about it are there. But, you know, that's a possibility, but it also depends on what you think.
Okay, thank you. So with limited time, I would like to go forward with the next one. So the next one is like we had discussion about the trainee QIA session that you would like to get answer from faculty members about your common question. But because of the time limitation during the boot camp, we didn't answer all questions. If you still feel like you want to get that question answered, please use this link and add your question, and I will try my best to share with everyone and get the feedback to you as far as I can get. So that will help you to, at least for some people that who are kind of stressful about the graduate life, to get a little bit not stressful. So moving forward with the next topic, this is just for your information. And the next one is about improving climate of BPRI. I would like Fabrizio to move forward and share our things that we are going to try best to help our trainees.
Yeah, so first I wanted to say that the session that you organized I think was great. It was really worked very well. And so one of the things that kind of came out was also that you know, there were some people that seemed like did not necessarily have very good relationship with their mentors. And so that obviously kind of is not good. So we definitely want you to be successful and to have good relationship and good interaction with your mentors. Now, it's difficult to know exactly what's the best way to solve this. My thinking is that, you know, the first strategy is always to kind of try to talk with your mentor if you can, so that you can clear up any kind of issues that might happen. And sometimes, you know, people just won't realize something, and so it's only good to do that if you can. But then if that's not really possible, then I think Jenna would be a good resource, and we kind of talked with her, and definitely she's completely outside of the, you know, of the whole trainee structure, and so she can definitely talk from an outside perspective and then give you know, her point of view, and then also talk with us while maintaining any confidentiality, because she's trained in that. So that's certainly a good way of doing things if you feel like that's the way to go. Otherwise, another possibility is always to talk with somebody else that is not necessarily your mentor, but that is in the BPRI. I think that's a great way of also of kind of discussing with other people. So you have to see whether you have kind of a good contact with another PI, or ask for it. I mean, that's something that could be, you know, we could find ways of assigning secondary mentors if you'd like that. I personally, when I was maybe not as a graduate student, a little bit less, but as a postdoc, I certainly had other mentors that were just not involved in my work, and that was really very useful to kind of decide what to do with my life and career and things like that. So that's definitely another way, and so that's more towards the geared towards also the scientific part of things. And otherwise, there's also a way. So I think Tanya also set up a way to send anonymous comments if that's a thing that you prefer. So there's a link here on the slide that she set up. So that's another way you can do this. But certainly, it's better for everyone if things work well, just because, you know, you're going to be more happy, but we're going to get better results, and so everybody certainly wants that, wants you to be successful, and the BPRI to be successful. And there's always going to be stress and difficult things to deal with, but the important is to kind of feel like, you know, you're really part of it, and that you can contribute the best you can. And so if you have anything you want us, also you can contact the executive committee or through the, you know, the student leadership council to kind of let us know, and we'll try to do our best to help. I don't know if there are any questions or remarks.
Hoju, do you want to add something?
No, I agree with everything that Fabrizio has said. That, you know, it's one of the things that we as a BPRI is striving for and very mindful of is the you know, developing climate that is inclusive, and so, you know, and then we're learning at the same time, and there has to be a constant communication and feedback so that we can improve. All right, so just like what Fabrizio has said, that our ultimate, of course, the research and everything that we're doing, we care about, but this is an institute and the training is a big part of it, and we want every single trainee to be successful, and that's what we care about the most. So I really hope that the trainees understand that, and then you know, we have means and resources to help out as much as possible, and so communicate. Right.
Okay, Spence, do you want to add something?
Well, I don't think I have much to add here. Did we want to talk about just sort of communication with the trainees with respect to the monthly meetings?
Yeah, so let's move into our next discussion topic, that how the cohort meeting will continue in next month, because we finish our all research presentation, each trainee presented. So what the topics that you would like to have and what the frequency you like to meet. We are going with monthly meeting frequency as we noted in our proposal, but if you feel it is kind of hard to manage with your time, we respect your time. So any opinion that you would like to change, so please free to add your suggestion.
And I think given that the trainees, we've learned through the first year, right, that trying to get everybody together for these can be a challenge. And so really it's important to, if you have some feedback or thoughts about how to improve the process, and it's the collective process. How do we reach out to you? How do we organize doodle polls? How do we minimize the number of emails that you get? Email fatigue is a real issue, right? And then, but keep in mind, one of the challenges, especially for Tanya, is that as she's trying to organize these, if she doesn't have sort of feedback from everybody, it's hard to sort of pick a date or pick a time. And so I think the other thing too, just in terms of, Ho-Jun's comments about being mindful about things, emails, right? We get a lot of them, we get overwhelmed with them, but I think with respect to these trainee meetings, if you can do your best to respond to doodle polls quickly, and I think, generally within sort of 24 to 48 hours. I had a PhD advisor that used to say NTLP, which was no time like the present, and sort of pushed us to sort of, do things when you could. And so keep that in mind. But Tanya's also aware of sort of reaching out to you and repeatedly ask you to fill out polls. So I think it's just do your best to respond to those. And when that doesn't happen, she will try to minimize reaching out to you, but it's part of her job to get these meetings organized. So I think the other thing too, then, is just, we don't have to decide today, sort of as Amanda said, sometimes it's good to step away and think about these things. Sort of, have maybe a discussion amongst yourself about frequencies, come to a consensus about what you guys like and sort of what fits best given your schedules. You're a year into the program now, you're a year into your research programs. And so hopefully you've got some ideas or thoughts, or if you think that things are generally okay, let us know that too. But if you do have ideas, please share them, especially with Tanya or the education committee, if you feel more comfortable doing that.
And adding following up, so I really need the what next month. Do you like to have like each PI coming and present their research, or do you like to have professional development topics that you especially interested? What kind of topic do you like to have in the cohort next meeting that really will benefit you guys?
One other sort of thought, I'll throw it out, and it's a bit of a rogue thought, but there is the Student Leadership Council, and maybe you guys need to meet occasionally as your own group, and maybe you're doing that already, but during that group, and when you get together and meet, all these topics can be discussed, and particularly ideas that you might have to streamline processes, make things work better for you, and that's an opportunity for you to have those conversations without people in leadership positions being present.
Please use the link if you like to comment anonymously and add your thoughts. So even I cannot recognize who commented that or who posted that one. So please feel to add that. And even after the meeting too, you can add your thoughts about that. I'm just going with the David idea that they like to learn about each ideas of the research, and we already had planned to have some kind of video series to introduce this research team. So until we get the kind of solid feedback from Leadership Council or you guys, I would say like next one, we can try to have one faculty member who is available to come and talk about their research until we figure out what's the best way to go. That is one of my suggestion in my side.
Yeah, I mean, that sort of goes in line with the one-on-one course idea that we did talk about. And, you know, I'm sorry that I was supposed to create something and I got so busy and I couldn't. And, you know, kind of making that sort of a lecture for me, well, it's not hard, but it's very one way. And I think that, you know, having actual interaction and students, you know, just ask questions so that I can, you know, answer or we can discuss. I think those are actually much more educational and informative than just one-way delivery of information. So I think that, you know, this could be something, you know, we can even record the whole session because it's going to be virtual, that if there are some topics that students are interested, you know, you can have a list of topics and we can have, you know, a particular PI who's comfortable talking about topics, commit, you know, one cohort meeting session and discuss that. There'll be, that's one way of kind of doing this sort of a, you know, workshop. If you I don't know, typically workshop does take more than, it's a long it takes more than one meeting, but you know, maybe there are subjects that can be covered within a relatively short time. So that's an idea. And another thing that I wanted to kind of say about the core meeting is that now that students, trainees have gone through, especially the graduate students have gone through their sort of first year, and you know, I think there has to be, it will be nice to have some sort of a, you know, interaction about how to progress through the degree, because you know, depending on school, you know, coursework might be, you know, still ongoing or off, and then there might be some sort of exam, depending on school, candidates exam, and all those things are coming up. And how do you kind of nicely progress through that that thing? Those steps will be, I think, hopefully useful to the trainees. And I say that because I talk about this to every single student of mine a lot. So and that means that students are curious, and I think it would be nice to have that sort of opportunity as well.
Thank you very much for sharing all the ideas. So before we are closer to ending our meeting time, so I wanted to give some quick overview about upcoming meeting. It is really important because we are trying to get all people into this meeting. So we will have BPRI plenary meeting. That is kind of a preparation meeting for the advisory committee meeting. It will be held in the September week of 12 to 16. We are still deciding time, and every trainee, we hope to see you there. And also we will have our second external advisory committee meeting. You may know that, some maybe don't know that we have four advisory, I think I'm correct, 4 advisory committee members for the research who are coming to our meeting and share about their research expertise. So we will meet them on October 4th 2:36. So you guys are all invited to attend that meeting. And also we will have like a special evaluation team. Me, Dr. Dooley, and Andrea will meet each of you individually to collect some feedback about the project progress and how you feel within the BPRI. And also the quick reminder for you guys that we will have the IDP review coming on the August and September. Please go back to your IDP plan and review where you are and what you haven't done and what you want to do next year. That is a quick reminder. I'm really like that I had this kind of opportunity when I'm doing PhD. I didn't had to someone to remind this kind of thing. So please go through with your all goals and review it and check it whether where you are at. Note that if you didn't do anything for that goal, this is completely fine. We are trying to get something done. So at least if you have done one, it is great. That is our plan. So thank you very much for your time. And please use Padlet to add your comments and suggestions. I will keep it open for like another 1 month. Then you can kind of brainstorm and have some ideas about our next meeting. And thank you very much for joining today.
Tanya, if I can stop you, can we sing you happy birthday really quickly?
How do you guys know that today is my birthday?
Yeah, so I just thought that we should just sing like all of us online to you from our respective places a happy birthday, if that is okay with everyone.
It's a great idea.
Thank you.
So on 3, so it's 1, 2, and 3.
Happy